Whole exome sequencing. Whole exome sequencing is a complete DNA test to help diagnose genetic disorders. Did you know that in every 1,000 live births, 10 individuals are affected by a genetic disorder? Most carriers of a genetic disorder have no family history of the disorder, and they do not know that they are at an increased risk of having an affected child. Genetic diseases are caused by changes in our genes. What does exome sequencing mean? Our bodies are made up of millions and millions of cells. All the cells in our bodies contain genetic material called DNA, which is packaged in structures called chromosomes. Our DNA is divided into thousands of genes, and our genes are the sets of instructions that tell our bodies how to grow and function. Each gene is used to make a protein, and proteins are what the cells use to perform their functions. A gene is made up of a sequence of letters in a particular order, and the 4 letters used to make this sequence are A, T, G, and C. Genetic testing, or sequencing, looks for changes in the sequence of letters in our genes that could cause disease, almost like looking for a spelling change. Some spelling changes are harmless, while others can affect the function of the gene and could be the cause of the condition in you or your child. Each gene is divided into sections called introns and exons, and the exons are the parts of the gene that are used to make the protein. Most of the spelling changes that are associated with a genetic condition occur in an exon. The exome is all of the exons in all of the genes in a person's body. Therefore, whole exome sequencing looks for spelling changes in all of the exons in all of the genes in a person's body. Single gene disorders can be classified by their inheritance patterns. Autosomal dominant disorders are caused by a variant in just one copy of a specific gene. Autosomal recessive disorders are caused by variants in both copies of a specific gene. Below you can see that an individual who carries one variant for a recessive disorder is called a carrier, while an individual with 2 variants will be at risk to develop the specific disorder. Most carriers of recessive disorders do not have family history of the disorder. In this picture, the individuals outlined in pink are healthy carriers of a recessive disorder. Carriers of recessive disorders are usually unaffected, but when 2 parents carry a variant in the same recessive gene, they have an increased risk of having a child affected with the disorder. If both parents have a variant in the same gene, the chance of having a child affected with that specific disorder is 25%. There is a 50% chance each child would be a carrier like their parents, and a 25% chance that each child will be an unaffected non-carrier. Many carriers of X-linked disorders do not have a family history of the disorder because family carriers are often unaffected. X-linked disorders are caused by a variant in a gene located on the X chromosome. Females typically have 2 X chromosomes, while males typically have 1 X chromosome and 1 Y chromosome. Females who carry a variant in a gene on the X chromosome are referred to as carriers and may be completely asymptomatic, depending on the specific disorder. In this picture, both females outlined in pink are carriers of an X-linked disorder. If a female carrier of an X-linked disorder has children, she will have a 25% chance to have a son affected with the disorder, a 25% chance to have a female carrier who, depending on the disorder, may or may not experience symptoms, a 25% chance to have an unaffected son, and a 25% chance to have an unaffected daughter. Whole exome sequencing allows us to assess if a person is a carrier of variants in certain genes. Whole exome sequencing is an important genetic test to consider when planning a family because it helps to determine the risk of having a child with certain genetic disorders, so that you can consider reproductive options to minimize that risk. Whole exome sequencing can help you plan for a healthy family. This test can help determine whether a couple is at risk of having a child affected by a common genetic disorder. If the parents have 1 or more variants in common, preventative measures can be taken in order to have a healthy child. This aspect of the whole exome test is recommended for consanguineous couples and any couples who would like to rule out the risk of having an affected child. Whole exome sequencing can also help identify disease-causing variants in individuals or families affected by a genetic disease. This aspect of whole exome sequencing is recommended for affected individuals who have a suspected genetic diagnosis, to help diagnose affected individuals who have multiple differential diagnoses, if targeted or panel testing was negative, and for couples who have a family history of a genetic disorder. A physician prescribes whole exome sequencing test that requires a simple blood draw. Once iGenomics receives your blood sample, the analysis is completed using an advanced technology, next-generation sequencing (NGS). A genetic report summarizing your results is generated so that your results can help you make more informed decisions during your reproductive journey. There are three main types of results that you can expect. 1. A positive result. This result would show that a spelling change was found that is the cause of the condition in you or your child. 2. A negative result. This result would mean that there are no spelling changes found in your DNA, which could be the cause of the condition in you or your child. 3. An uncertain result. This means that a spelling change has been found, but it is unclear whether this is the cause of the condition in you or your child, or just a harmless spelling change. Incidental findings. Because this genetic test looks at all the exons present in your DNA, there is a chance that the scientists could find a spelling change in your DNA that is not related to the condition present in you or your child. This type of change is called an incidental finding, where the test finds a spelling change that is associated with a different condition than the specific condition for which a person is being tested. Genetic counseling is recommended before and after genetic testing. You can also request a genetic counseling session that will help you better understand your test results while you make an informed decision about your next steps. Get your free genetic counseling session with iGenomics expert genetic counselors. Call 971-800-50342 or email info.me@igenomics.com. iGenomics, with science on your side.